Well, good morning, everyone. I just want to say thank you for joining us for today's virtual Lunch and Learn workshop. This is the first of a 4-week series of Lunch and Learns. If you have enough bandwidth, we'd love to see your smiling faces with your cameras on, especially at the end when there's time for questions and answers. If you have any questions during the presentation, please take a minute and just type it in the chat. If it's relevant to what we're speaking about at the time, we may answer that question right away. Otherwise, we'll save them for the end. And I think that's all I have for the introduction. So this is, I'm Melissa Rushaw. I work for the Du Bois County Soil and Water Conservation District. I specifically work on the project at Vincennes University Jasper campus, the Land Stewardship Initiative, and this is my co-presenter, Stephanie.
Good morning, everyone. My name is Stephanie McLean. I'm the Soil Health Specialist for the Indiana Natural Resources Conservation Service. So my work focuses on soil health education and outreach, focusing specifically on management practices that landowners can do to improve soil health on their cropland, and then working with partners like Melissa and the LSI and the Du Bois SWCD and the conservation partners within our agency to improve that outreach to our farmers and help them have success in implementing those conservation practices. So Melissa, I'm running the PowerPoint today, so I'm going to move this forward, but we are going to kind of tag team a little bit of our presentation as we go through the session here. If I'm sitting on a slide too long, Melissa, and you're ready to switch the slide, say next slide, and I will do that, okay?
Fair enough.
All right, next slide. So we decided to start this this first session off, this like inaugural kickoff for this Lunch and Learn session, these sessions that the LSI team has developed to really get back down into the basics of soil health and what that definition means when we talk about soil health on ag land, so that as you're out looking at your farms and in your fields, you can really think about this definition and how it applies to you on your land. So when we talk about soil health, we're talking about the continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. And that's a really big mouthful of a definition. And a few things I want to unpack here before Melissa takes us into the really nitty-gritty details, the most basic of the details that we really want people to remember. But let's unpack some of this definition here. The first thing that I want to unpack is the phrase continued capacity. That idea that when we have a healthy soil, when we have soil function, that a soil can continue to do it's continually able to do its job. Even if something happens, there's maybe an event that occurs where you see erosion or runoff or other things like that on a generally healthy soil, but over time that soil is able to rebuild itself. So again, that continued capacity, it's not just a one and done thing, it's constantly occurring and continually repairing and rebuilding itself. The next thing there is the definition, or thinking about function, and function in its most purest sense is thinking about what a soil is supposed to do. Just like when we think about, how our bodies are supposed to function. When you go to a doctor and the doctor says something's not right, it usually has to do with something not functioning properly, right? Something's broken or something's out of whack. And the same is for our soils. There's a given set of functions that our soil provides and does, and it's just supposed to do that. And when it isn't happening, we have a loss of function on our landscape. And so we think about function as the ability of a soil to infiltrate water, the ability of a soil to be physically stable and support traffic and weight. Our soil, when it's functioning well, is a home for organisms that live in the soil. And another key function is nutrient cycling. And all of these things, when they're all together, the ultimate function is resiliency in that system. And so we really want to think about those functions and that soil is able to do these things, right? The things that it's supposed to do. And typically we think about functions and we notice them when we don't have them in those systems. So that's the next thing we want to think about. And then lastly is this idea of living ecosystems. The fact that, we don't think about it very much, but our soil is a living, breathing ecosystem. There's a ton of people out there that went to school for wildlife biology and all these other things. And they talk about systems and ecosystems and how, when we have a high rabbit population, the wolf population increases to consume the rabbits, and that whole system approach of thinking about things. And guess what? It's exactly the same in our soils. There's a whole ecosystem, living, breathing ecosystem down there that functions in that same way. And then, the last idea is that ecosystem is able to sustain the plants, the animals, and the humans. And I really like to think about it even more, not just sustaining us on some level playing field, right? But regenerating us, improving things over time when we have a healthy functioning soil. So that was a lot to unpack there, but those are the things that I really like to think about when I talk about soil health. And Melissa, this is the great slide that kind of coins it all.
Yeah, this is a slide that I enjoy, probably because I'm from that biology background. But what I like to look at soil health is kind of that sweet spot in the middle where the physical characteristics, the chemical characteristics, and the biological characteristics of the soil are working together with each other and functioning well. Your physical soil is what you have. It's the sand, the silt, clay makeup of the soil. You can change the physical structure a little bit with infiltration, but basically kind of what you have is what you have. Chemicals is something that can be altered. Obviously, when we apply nutrients to our soil, we are changing the chemical balance in the soil, and we want to make sure that's in balance with what can handle. And you need that diversity in the soil. And we'll come back to this later when we look at some other slides, but you need a variety and diversity of biological organisms in the soil to function and make it healthy. So next slide.
And to kind of complement on top of what Melissa said there is that, and we're going to touch back more on this later with that biological piece of this Venn diagram, but that biological piece of the puzzle is responsible for 90% of the function that occurs in our soil. So although it's just 1/3 of that one leg of that three-legged stool, or however else you want to think about it, that biological aspect is extremely key in building and maintaining that function and supporting all of the other things that Melissa highlighted there.
So there are things that we can do that kind of put us on a downward spiral and not help to degrade the soil, but cause the soil to become degraded. Tillage and lack of residue will cause the soil organic matter to go down, erosion to increase, impacts to increase. As that's happening, the aggregates break down and it's not a stable soil. So you're going to see crusting on the surface and compaction issues, which is going to lead to a loss of infiltration. When you have that crust, you cannot get water into the soil, and you're going to see more soil carried off with erosion. If you have lower organic matter, you don't have the ability for the plants to have those nutrients that it can take up, leading to ponding and other issues as well as disease.
And all of it kind of, this diagram kind of steps through this. They call it the downward spiral of soil degradation, but it really does hit on a lot of things that we tend to see in our soil landscape, right? Where we see excessive amounts of ponding after a rainfall event, or again, that water's not getting in, so we have runoff and erosion. And a lot of that all starts with that physical disturbance that occurs on our soil and then we start to lose our organic matter and those aggregates, which are essential for all of these things that we look at here. Our water stable aggregates start to break down, and then that whole thing just kind of collapses like a row of dominoes. So
this past summer, I collected some samples from one of our fields, field 2. It's a place where we have a tilled strip down the middle so that we can compare side by side how our soil is functioning as tilled soil, and then the rest of the farm has no-till soil with cover crops. So these are the 2 samples that we're going to put in a Slake tube, and we're going to start running a video here in just a second. And you're going to see the tilled sample on the left and the no-till on the right. And I went ahead and cut the video to the first minute, because in the first minute you see me dropping the samples in, And it takes a little while for all the water to rush into the sample and start taking up the pore spaces that the air has and filling it with water. But this is what's going to happen here.
All right, Melissa, I'm going to hit the go button.
All right. So as you can see, after 1 minute, you're starting to have particles of soil, falling and breaking apart. And in about 5 seconds, the sample on the left is going to shift, and we're going to get a big explosion of soil. And if you're looking on the right, it's stable. It's not dropping pieces of soil. It's staying stable and together because it has those roots in there. It has the glomalin, the sugars holding it together, which when you till the soil, you're breaking those up, and so it's losing some function. If you have a big enough screen and you look closely enough, you can see air bubbles bubbling up out of the sample on the left. That's just as the air's coming out and the water's rushing in and causing it to fall apart. You can see the water in the tube is a darker color. It's a little difficult to tell, but there's suspended particles of soil in that tube, and you're starting to see at the bottom there, how much soil has already fallen away. Anything you want to add to that, Stephanie?
Nope, I clicked it. Maybe I should have waited a second, but I think one of the things that's really key to think about there is that for us to have, when we do that demonstration and we put those soils in there, it's what's really essential about that, for that to really show things, is that water wants to rush into that soil. And that soil on the right that Melissa had, that came from that one part of the LSI site where they had more cover crops and no-till, that those pore spaces are well maintained and that water is able to rush into it and push that air out. Whereas on the other side, that left side, the one that was falling apart, again, that water is trying to rush in and it's not having success getting into those pore spaces because they're not continuous. There's no continuity to them. And so then what happens is that as that water is trying to push in, the air is trapped in there and it explodes to get out. And that's why we're seeing those things happen when we look at those slakes. And what's really cool about that demonstration is that those were collected again on the same field with different management, with what, 10 years or less of different management. So you can see some significant changes in the ability of a soil to function or not function in a pretty short period of time. So if you want to kind of talk about the LSI, Melissa, that would be great.
Yeah. So this is just an overview shot of the Land Stewardship Initiative. You can see 5 different sizes of farm fields right there, literally on campus. Go ahead and switch to the next slide, Stephanie. So as Alan mentioned in the chat just a second ago, that we started cropping this in 2012. That's when we started the tilled strip in that center of field 2, as well as farming the rest of it with no-till and cover crops. So these samples are we have that blue box there that's crossing over 2 grassed waterways. There are a lot of best management practices that have been utilized on a campus just to help keep the soil stable. A series of wasp cobs at the top of field 2, several different grass waterways, French drains. We have a 40-foot wide strip down the center that is tilled every year, and we'll show pictures of that later. So we use that to be able to compare side by side, same soil type, different management practices. What are the differences in the soil? Go ahead to the next slide.
All right.
So this past fall, Stephanie was able to come out to the farm.
Did it work, Melissa, or did I hit the wrong slide?
Nope, we're good. Sampling at LSI. So nope, we backtracked there. There we go. So here we are. That's Stephanie digging in some soil. That's not LSI, but it was a good background for letting us know that we're sampling here. So moving on to the next one.
So one of the things that I know that Melissa has sampled for and is kind of more of a new sampling procedure here at LSI is looking again for those water stable aggregates, and we've talked a little bit about water stable aggregates, but like I said, they're so essential and really important to think about that. That using the demonstration or using an assessment of looking at water stable aggregates is one of the first things that I would recommend when going out to a field site. So when we talk about managing for water stable aggregates, if you look at that picture on the left side where we're seeing infiltration, right? Water is supposed to infiltrate between the pore spaces of the soil aggregates. We want that water to go into the soil where it lands, if at all possible. Water shouldn't have to flow across the surface of the soil to find, say, an earthworm channel or other things like that. Although earthworm channels are really essential for, say, sudden downpours, massive amounts of rainfall, where those larger channels can take in a lot of water all at once. But really, the most important aspect of infiltration is that water soaking through those pore spaces and going into the soil where it lands. By way of comparison, when we look at that other side there on the right-hand side, when we don't have water-stable aggregates, those aggregates will actually start to melt and fall apart and then spill up at the soil surface, like you see here in this picture. And so that's something that we don't want to have, because when we have those water stable aggregates melt and fall apart at the soil surface, we get runoff. And so what we're going to do now is talk a little bit about the slump assessment and how you can use that for assessing, again, water stable aggregates on your field. We're just kind of looking at some of the pictures here as we talk about slumps. So I'm going to stop my sharing just a second here, and I'm going to do the demonstration, and then Melissa's going to talk about the times that we were out in the field this summer at the LSI site and what we saw while we were out there. All right. I'm going to turn off this camera, and we're going to turn on the other camera. Okay. All right. Melissa, let me know. Can you see everything okay?
Yes, that's perfect.
All right.
We're
So
not sideways this time.
yes, technical difficulties. They're always such a joy to deal with. All right, so I have two soils that are here. I collected these, and I'm going to use them for the demonstrations. But they are soils that come from southwestern Indiana. Maybe I can't remember exactly in the county where, in Du Bois County, if they're one of the counties right next door. But I'm going to kind of bring this soil up, and I'm actually going to tilt the camera down so you guys can get a really good look at this soil. Right, so remember we talked about those aggregates right here? So we're really going to look at these aggregates and the key role that they play in allowing infiltration. So when we think about what an aggregate is, it's a conglomeration of sand, silts, and clay particles, organic matter, and then plant roots, microbes, fungi and fungal hyphae, and all of those things wrapped together. So when you get an aggregate like this, we're going to pick this one up and just kind of focus on this a second. When you get an aggregate like this, you have all of these things holding those soil particles together. And when you break it up, it breaks into smaller and smaller aggregates over time. So it's really holding itself together in a way that really allows those aggregates to hold together. So those are the aggregates that we're talking about when we talk about water stable aggregate. It's not just soil particles, but it's all of those things being held together with those plant roots, with that organic matter, with the glomalin that's being exuded from the microbes that live in the soil, coating those things and holding everything together. And what we create, and what we can't take credit for this, what those microbes create is a home, a functional ecosystem that is their home, that then we as humans with our plants and the things that we do in the landscape benefit from that, because that home is not just a stable ecosystem for those microbes to live in, but it's a stable ecosystem for which our plants are able to grow in as well. So again, this soil comes from a field that's been in a long-term no-till system, 25 years or more, and then we also have had cover crops every year on these fields since for the last 10 or 12 years. This soil over here, again, is same soil type, but comes from a different management system, and we've seen a considerable amount more erosion on this field. See that physical differences, their colors are quite different, right? But also when we start to look at these aggregates that I mentioned before, and we start to break them up, I'm going to kind of open one up here a second, and we start to look at it a little bit, we don't see as many spaces and other things in there. And as we open them up, what starts to happen is that they actually kind of fall apart to their individual particles of sand, silt, and clay. Because that soil is so overworked, right? The glues that are in there from the microbes, they're not able to hold together because they're constantly being stirred and broken down. And those, again, those aggregates that we talk about with the plant roots and the root channels and the microbes and all of those things kind of holding everything together, those things are constantly being destroyed from that physical disturbance. And so we end up having aggregates that I would call weak, that when they open up, they fall apart and they crumble. So what we're going to do is we're actually going to take these soils and we're going to put some cups of water, which you can see right into there. So I'm just going to take some of this soil. I'm going to just dump it right in here if you guys can kind of see what I'm doing. Right, and then we'll do this for the next one. Get a little bit more in there. And then we're going to just take these and we're going to stick them in the water. And then we're going to kind of look at what happens to these, soils when we put them in the water. I'm going to put some plates down right here, and I'm going to set these trays on here so you can see them. All right, so I'm guessing you guys can already see a difference, right? Right away when we put those in the water, again, that soil that came from that more stable ecosystem, right? Less disturbance, more cover crops, more protection for that living ecosystem. When I put those in the water looks pretty clean in there, right? And this one over here, when we look at this in the water, again, it looks very similar to that Slake test that Melissa had, right? That soil's kind of going into suspension. We're seeing that water turn cloudy because, again, those particles of sand, silt, and clay, in particular, that clay is hanging in suspension in that water. So we're kind of getting a glimpse of what's happening in that tray as we water through those aggregates. Remember, we said we want that water to flow freely through those aggregates, and those aggregates we want to hold together. So now we're going to take them out of these strainers and see what they look like on the plate. And I'll do these at the same time. I'm going to just kind of make sure everything's right in the middle of the screen here. Take some time for that water to drip out just a minute. And then we'll tip them over onto the plate. We'll take a serious look at these things. I'm actually going to put the camera a little bit closer and turn it down onto the there we go. All right, so the first thing that we really see here, I'm going to bring this one closer here. We can still see those aggregates, right? You guys can kind of see the shape of those aggregates being held in there yet. Even though they got wet, they still have that angular pointy shape. We can see a little bit of water coming out of it, right? But most of that water is being held in that slump because of the organic matter, because of the pore spaces, because all of those things that have been built in that soil to hold that water in there. The other soil here, again, we aren't really seeing any of those aggregates anymore. Anything that was in there that was considered an aggregate, it melted when it got wet. So that melting has, again, as those aggregates melt, then what we're left at the surface of the soil is this mud pie type consistency. And again, when this mud pie dries up, what we're left with is a soil that is more likely to be crusted when it's dry. We also see quite a bit of water coming out of this slump as well. Again, when water comes out of the slump, and a little bit came out of the other one, that's just an indication that there's no pore spaces for that water to be held in, and there's no organic matter holding that water in that slump. The last thing that I like to do when I look at these is I like to slide them on the plate and see how easy it is for that slump to actually slide across the plate. So you see how that slump is sliding across the plate like that? It's not holding together. It's not holding water. It's not.
Sounds like Stephanie's voice cut out there.
So we are gonna, I'm gonna stop this video and we're gonna go back to the PowerPoint presentation. Let's see here. I got to share my screen a second. I think. There we go. All right. Am I sharing the PowerPoint screen, Melissa?
Not the individual slides. You're sharing the whole thing.
Okay. Hold on. It always just takes a little bit of time to. It always takes a little bit of time for the magic to happen. So again,
through this. Sorry.
That's okay. We were talking about those slumps again and looking at what that soil looks like when we go through that assessment. And you can see some different examples here of this picture, right? Like in this shovel here, the soil on the right has more water stable aggregates than the soil on the left, which is really melted apart. The soil up on the top, we just see that water oozing out of that slump. And we also see how shiny it looks over the surface of that slump. All right, Melissa, you want to talk about the about some of the stuff that we did this fall out there?
So these are pictures. That's actually Stephanie holding the slump demos that we did at LSI. And if you're looking, it looks just like what she just did on film. The slump on the left is from the tilled soil that, again, since 2012 has been continuously tilled down that strip. And the soil on the right has that stability. It's holding the water into it. It's our no-till cover crop soil. And that's just in 9 years. We've changed the color of the soil. It all would have been what was on the left if we had not changed the management practice. So we're now looking at a much healthier soil, a much more functioning soil, a healthier soil. So I was listening to a webinar over this past year, and I think it was Bob Ward who was talking about how important infiltration is. And he said, it doesn't matter how much rain you get, it matters how much rain you kept. So if you get a 4-inch rain and only that first inch is going into the soil and the next 3 are running off, you did not get 4 inches of rain. You still got 1 inch of rain. So this next series of slides is something called a rain infiltration ring. And so you pound this into the soil straight down about 3 inches, and then you put a layer of plastic there, and you put in 1 inch of water, and you pull the plastic away, and you time how long it takes for the water to infiltrate into the soil. And so at LSI, the first inch of water to infiltrate into the soil takes about 15 minutes. So if we have a light rain, we're golden. And it's similar between the tilled and the no-tilled. It's gone as quickly as 5 minutes, and it's taken as long as 25, but the average is about 15 minutes for the soil and Land Stewardship Initiative to infiltrate that first inch. The second inch is where it gets a little trickier. That second inch still takes a little over an hour in the no-till area, but in that tilled strip, it's better part of 1 hour and 47 minutes, over 2 hours. There's times I've had to walk away from it because I need to go pick my kids up from school. I picked up a trick from Stephanie where you can just measure how far down it's infiltrated, and just measure the depth of water left in the ring, and then extrapolate how long it would still take to do that. But the next slide shows the samples we did back in October when she was at the field. So go ahead. And again, you can see the color differences in the soil. You have this, the sample on the left is the tilled area. We're trying to keep consistent with putting them always on the left, and the no-till on the right. You can see differences in color. You can see worm channels on the right. And the timings were very similar, but I'll let Stephanie go ahead and talk about the red area there. She added a picture in because it's hard to tell on our screens, but that soil was still dry. The water didn't infiltrate into the whole ring. It had to, whether that's a compaction issue, well, likely that's probably what it is, but the water could not infiltrate through that whole surface of the ring. It only got to go down one side.
Yep, great point, Melissa. And I think that's something that we really want to think about when Melissa's out doing these infiltration checks and things like that. It's really dependent. It's really key that there's consistency in how the ring is pounded in, and thinking like Melissa said, how dry is the soil before you rain on it, and all of that stuff like that. And that's why when this is used, say, as an assessment on a site, you want to always try to measure that second inch of rain if possible. But if you look at that soil on the right, you can see the whole surface of that soil. And again, it's not the surface, it's the bottom of a 3-inch infiltration ring. But that whole surface of that soil there is shining, right? And it's got a sheen to it when the sun reflects on it, which is telling us that that water is infiltrating throughout that whole entire soil profile. We have those macro pores that Melissa pointed out, those earthworm channels, maybe some leftover root channels. But again, we're having that infiltration through that soil matrix overall. And by way of comparison, on the left side here, just where that that red's marking went up, when you look at that picture, the top side of that, it's shiny. There's infiltration there. And we're actually even seeing some possible root channels, some earthworm channels, different things like that. But on this half over here, on the bottom half, there is no infiltration that's occurring there. It's dry. And so again, is infiltration on only 50% of your field acceptable? I would hope not. And so when we think about that, again, those water stable aggregates and the ability of a soil to infiltrate, we want all of your water to fall on your field and 100% of it to soak in where it lands. So one of the reasons why, or one of the factors that play a big role in as to why some of these things are happening out there is again pore spaces, root channels, earthworm channels, those pore spaces. So we really want to think about what a healthy soil has, or what a functioning soil has, in comparison to, say, a soil that is not healthy and functioning. And so these things that we talked about here, we want to think about those pores that are in our soil as the lungs and the circulatory system. Remember, I said that this that soil is a living, breathing ecosystem. Those organisms that are in the soil that are breaking down our residues, that are putting those glues and coating those aggregates, they're aerobic. They need oxygen to breathe. So saturated soils with a crusted surface that air can't exchange freely into the soil, those are soils that are losing function and that don't have good lungs and circulatory system. And not only are those pores and that lung and circulatory system important for airflow, it's also essential for that water flow and storage and availability throughout the growing seasons. So how does that compare to that compacted soil? And if you look at that pie chart, you know, when a soil gets compacted, whether that's, again, trafficking or being worked over and over again and trafficked over, it's not that you actually gain more soil here, right? It's that you're actually losing pore spaces, that volume of soil, the same volume of soil, is going to have less ability for air and water to be in it. And so that's really important to think about as we want to focus on that function coming back into our soil ecosystem. Anything to add, Melissa, before we flip?
Nope, looks good to me because we're starting to run a little low on time.
All right, compaction. That was something else that we looked out there at the site. And we want to just think about, compacting layers. This is a great picture of some compaction that I found in another field where I dug it up, and you can see layers over time, compaction from traffic.
So this next picture, this is looking down the tilled strip. You can see that we are growing cover crops in the no-till cover crop area, and we did not plant cover crops in the tilled strip. However, there is some green there. Nature abhors a vacuum, so you're going to have henbit, purple dead nettle, and different things growing in there just because the soil, they're going to grow somewhere. But it's probably a better choice to choose the cover crop you want to have growing so you can manage for it. The next picture, in case you're wondering what it looks like when we're disking, that's Bart disking the soil for us. That's just the very first pass on the very first edge, but it ends up looking even and brown by the time he's completely done.
How many times do you think, like in a typical system, Melissa, for working over 2 times, 3 times disked before planting?
I honestly don't know. He worked it till he felt it was mellow enough. And I wish I'd share the picture of what it looks like when it's done, but it's brown. You don't see any residue left. All the residue is completely buried. That's pretty much his goal, get it all down. So. So these are pictures, again, taken from LSI when Stephanie was out there. She used a flat spade shovel to dig down. And then she kind of just she peeled this off the edge of the hole and just kind of dropped it flat on the ground, which caused it to kind of shatter along some of those compaction zones. Okay, Alan says the disc strip has about 3 passes of the disc. Thank you, Alan. I knew it was several. I knew it took a little while, but thank you for the good number. So this is a picture of some of those compaction zones where it's going to split, where the surface of the soils are making planes.
And this is another assessment that Melissa does out there with the penetrometer and just looking at those layers to see, how deep they are and other things like that. And now we can look at the comparison.
And at what depth do you get that root restricting zone where the roots are just gonna have to go sideways because they can't go down any further. So again, side-by-side shot of tilled strip on the left, no till on the right. And Stephanie's going to overlay that with some, and you can even see some different lines in there. If you're looking closely, there's quite a few more in the tilled strip.
Yeah, when we look at these lines, these horizontal fracture zones, there's a lot of different reasons why we can have these in our soil, and understanding, how restrictive they are, and also, again, trying to piece into why they are there in the first place is a really key important part of understanding how to bring function back or how to rebuild a healthy soil. So again, that soil on the left, what we're looking at is we're seeing multiple compaction zones or multiple horizontal fracturing layers. And the ones that I really want to key in on are the 2 that are in the middle. Those are probably at about, I'd say 4 to 6 inches down. So it'd probably be some of the resulting from the spring tillage passes that are there. And then the other ones that I'm really interested in are those layers up on the top. Do we notice those layers? And I would guess that I was talking to Melissa and I asked her if there was vertical tillage, some type of surface tillage that is occurring on the site, because that would be very typical of a VT pass, that disturbance and then those layers getting formed with the VT pass. And she had said no, but one of the things that made me think about is again, a bare soil with a heavy rainfall event and weak aggregates. We talked about that crusting. And if that happens over and over and over again, you can build up layers of a crust that are maybe 1/8 to 1/4 inch thick, and then you can build another crust on top of it, and you can build another crust on top of that as that soil continues to degrade and fall apart. And think about how hard it would be for water to infiltrate into that soil after those crusting events have occurred, or those events have occurred where those crusts are formed. And then on the other side, again, we're still seeing some horizontal fracturing. And you have to remember that compaction doesn't go away overnight. It took hundreds of years of farming these lands and for those aggregates to break down, for us to really start to notice our loss of function in our soils. And so we can't expect that damage to be undone overnight. But what I was really interested in with this one is the fact that there's only one horizontal fracture at that 4 to 6 inch range right there. And so we're also, what we're seeing is that soil really has such a great texture to it, that structure. We're seeing the whole, like the soil looks very granular. Again, we talked about all those aggregates. And compared to the other one that almost kind of has like a massive look about it, right? There's not a lot of individual soil aggregates in there. So again, this soil that's on the right has really, it's really changed its physical shape and structure and color. And again, that's because that soil function has come back to that site. All right, Melissa, life in the soil. This is, I think, your favorite part.
Yeah, I'm a biology major. There were 2 types of biology majors at our school. There were the campfire biologists, which I was one, or the cell smashers. This is a little bit more on the cell smashing stuff, but I enjoy looking at the big picture. So just to help me understand the scale of what I was looking at under the microscope, I borrowed some of my daughter's playground sand and just put that under the microscope so I could understand how small the organisms were that were living in the soil at that scale. So this is grains of sand magnified 100 times. If I were to magnify it 400 times, it would fill the entire slide. Next. So we found quite a few different things, most of which did not make it to this slide set, but there are things that you can sometimes even see with your naked eye. So if you're looking at the left of this slide, you're seeing a fungal hyphae, and you can see, and that's minerals behind it. Those are not pieces of sand, those are just further broken down, like the silt and the clay and other minerals, but you can see the cell walls. in that fungus, so you know that's what that is. Kind of like a plant root here over here with some materials gathered onto it. And then down here, again, it's zoomed, you kind of ended up small, but there's bacteria in the soil. And they are living right on the surface of those aggregates. They are not living in those pore spaces. And they, what Jim Horman likes to call, they're the soluble bags of fertilizer. They're the ones that are digesting the nitrogen that you're putting onto the soil and making it and turning it into a plant usable form. So these are critical to have as part of your soil. They'll encapsulate themselves when it's too hot or they're too cold, but when they're growing and flourishing, they're reproducing at a rapid rate, and they are being eaten by the next slide. So this is a Protozoa Cilia. His job is to move around in the soil and eat the bacteria and digest them, releasing those chemicals that the plants can use. So I don't know if it's going to start playing or not. So again, this is at 400 times. You can see some smaller bacteria in the background. You can see some other just particles of dirt. But this is another part of that whole web food chain. You have things like nematodes and other things that are even bigger predators and worms that you can obviously see those with your naked eye, but these are the ones you can't see that are critical. Go ahead on.
Can you see it moving, Melissa?
Yes, I can see it moving.
Wonderful. So Melissa kind of touched a little bit briefly on, again, those organisms that are so important for building that ecosystem. And this just kind of touches on that even to a greater scale for us to understand the amount of bacteria and fungi and even algae in a soil situation that is living and thriving in a healthy functioning soil. And so it's just so important for us to think about all of these things that are in there. And I know so often the question goes back to, say, more of a problem fungi or a pest or other things that we have out there. And the biggest thing that you want to keep in mind is that that's just one or two of these organisms in comparison to millions upon millions of other organisms that are living there, hundreds of different species that are outnumbering that one pest. And so the key in building a healthy functioning soil is to increase the number of the beneficial organisms, the microbes, and the other microbes that are in there just working for you, so that the pests are outnumbered. If they're 1 in 1,000, then their impact is going to be so much more limited in comparison to if all of the other beneficials and hardworking organisms have been killed off, or they're dormant, like Melissa said, because conditions aren't right for them to grow. Then the ones that are in there that are pests are going to thrive and take over the system. And so we really want to think about that balance and thinking about how our soils are a host to, again, a vast number and mass of organisms. And by mass, it's like it shows here, approximately the equivalent weight of 20 cows on a hectare of soil. So in 2.5 acres, the biomass of just the living organisms, that's not soil organic matter, that's not the dead materials or the residues, that's just the living organisms that are underneath the soil, is the equivalent of 20 cows. Think about how amazing that is and how much work those things are actually, living and doing and thriving in that soil to make that whole system function.
So now if we were at a normal workshop, we could go back to the Slake test and show you what it's done. And so here we are. This is a picture from an hour later. And what I like about this is if you look on the right, you can see those microaggregates being held on by the roots. The water is still cloudy, and that tilled strip, yeah, thank you, Stephanie. So there's microaggregates hanging. There's actually probably even some microaggregates hanging on the left hand too, but the one on the right will stick around. So even after one whole day, those microaggregates are still holding onto those roots. That soil is still stable. Whereas on the left, it's just about completely gone. And if we look towards the bottom of the tubes, you can see just how much soil particles have fallen away, and you can even see those like mini compaction layers as a layer of soil fell away, and then just as the water settled out. So again, that one on the left is from the tilled soil. The one on the right is from the no-till soil with cover crops. You can see the pieces. So those were the original samples on top, with the worm channels and everything. And it's amazing how much you can lose, the potential to lose when you're tilling. So I guess that's my shot of a week later. I don't recommend going out a week because the soil gets anaerobic and smells terrible when you're trying to clean it up. But I mean, look at this. This is still a week later and it's still holding together. So in the very beginning of the presentation, we kind of showed things that you do that are done to the soil that break it down, cause it to lose its function. This next slide is the soil aggregation climb. So when you stop tilling, you start doing those cover crops and those things. These are things that will happen to your soil. The biological activity will increase. So you're going to get more organic matter, which is going to improve the nutrient cycling, which is going to improve the cell structure, which is going to improve the water availability and the permeability of it, giving you a healthier soil.
And what's really important to think about with this is, as you're looking at taking this journey, this soil health journey on your farm, you want to think about where you are on the stair-step approach or this process. Again, if soil biology is essential for rebuilding soil aggregates and improving soil function, then we got to start by bringing those guys back, and gals, of course. But as we add, say, cover crops to a system, we're going to notice right away increased infiltration, right? Because we're protecting that soil surface and we're going to have less erosion. But we really got to think about that biological activity and that organic matter turnover as the capstones or as the basis for improving nutrient cycling and improving structure. And what I like about this slide, again, is that a lot of the things that we talk about, they're so important to your operation and why you farm and how you farm. And so there's a lot of, say, feeling and why associated with it, but there's also research that backs these things. And so, as you're talking to other people or you're thinking about some of these changes, know that there's a ton of research out there that is showing these changes are occurring. And we've listed this article from Crops and Soils magazine from Jerry Hatfield that has actually showed, research and it has proven over and over again, increased organic matter, holds more water, keeps soils and plants more productive during droughts times of the year. And then again, that idea of just increasing that organic matter and improving that soil is the way that we want to build and improve function in that soil ecosystem. So I think that's probably all we have for you today. Melissa, is there anything else that we want to touch on, or is there any questions that we want to start focusing on?
Well, yeah, does anyone have any questions? You're welcome to turn your video on and your camera on, or you can type it in the chat.
And I will stop sharing the screen so that way I can actually get into more of like that gallery mode. So Melissa, it looks like we have a couple chats here. I'm not sure what they are. Okay.
Those are from Alan, just commenting on things that we weren't sure on. The 3 passes, right. And then he had mentioned the 2012 before I got to it. Well, we'd love for everyone to join us again next week. We have some local farmers / ag retailers, professionals who've been helping us with they're partners with us on the Land Stewardship Initiative project. And they're going to discuss their own personal journey in soil health. And they're going to share some of the changes they've seen on the farm and their time working with it. I know some of the things that have been measured are that organic matter and that cation exchange capacity. And we'll be talking about those next week. That's not something a farmers can go out there with a shovel or with a little sink strainer and measure. That's something you need to take the sample and send it to the lab. So that we did not touch on those this week. But that organic matter has increased, and you can find out how much next week.
That's a nice cliffhanger, Melissa.
Yeah,
I like that. You got to keep them coming back. Like, find out, check in next week. But Melissa, there is a question here from wondering if the samples that were used in the Slake demo that you recorded, did those soils come from LSI?
Yes, they did, Alan. They were taken. We tilled in April. I harvested those in July, which is why I think there was maybe a little bit of roots in there, as I was expecting it to explode a lot more. But yes, those were taken directly from the fields. And I still have bags of those samples, so we can do it again anytime we need to. So yes, those were taken from the fields, side-by-side comparison, same soil, different management from LSI. The slump test that Stephanie did was not our soil, but the Slake was.
And the pictures that we had later of the slump,
that was the same.
That was from LSI, yeah. But I just wanted to try to do a hands-on demo, a little bit of an interactive process as we do these virtual sessions. It's kind of nice to break from a PowerPoint, have a video like Melissa did, then break from the PowerPoint, and then kind of do a little live interactive session. It really keeps people engaged, you know, as we try to do some different things here and work with the attendees to get them to think about soil and soil function and what's going on their farms.
Yes.
Does anybody have any other questions or any other things that we want to discuss? Melissa, is there anything else that we want to talk about that we think about with the LSI?
I was gonna say, it looks like Alan just invited anyone to come out and have a close look at the soils on LSI. Yeah, please just give the Du Bois County Soil and Water Conservation District office a call or shoot me an email. My information was on the previous slide, but you can look me up on Facebook or on the web. It's Du Bois County Soil and Water Conservation District. And Alan or I or any of the people partnered with the project would be glad to take you out there, show you some of the things we put in, show you the French drains, show you the field borders, you know, the functional wasp cobs. We can go out there with a shovel and some of these things and look at our soil. We can come to your farm and look at your soil, see where you're at on your soil health journey. But again, that's a part of week 4. So great, keep tuning in, guys.